General Biology, Part 02. Breathing. Breathing is the physical process of exchanging gases between the lungs and the atmosphere. It is a two-step process, inhalation and exhalation. During inhalation, the diaphragm contracts and the lungs expand, drawing air into the lungs. During exhalation, the diaphragm relaxes and the lungs contract, pushing air out of the lungs. Human inhales O2, exhales CO2. When we breathe in, we take in oxygen, O2. This oxygen is then transported by the blood to the cells of the body. The cells use the oxygen to break down glucose and release energy. This process is called respiration. As a byproduct of respiration, carbon dioxide, CO2, is produced. The CO2 is then transported by the blood to the lungs and exhaled. Plant uses CO2, releases O2. Plants also need oxygen for respiration. However, plants also produce oxygen through a process called photosynthesis. Photosynthesis is the process by which plants use sunlight to convert carbon dioxide and water into glucose and oxygen. The oxygen produced by photosynthesis is released into the atmosphere, where it can be breathed in by humans and other animals. Respiration. Respiration is the chemical process of using oxygen to break down glucose and release energy. It takes place in the cells of the body. During respiration, oxygen is taken in from the lungs and used to combine with glucose to form carbon dioxide and water. The energy released from this reaction is used by the cells to carry out their functions. Respiration. Oxidation of glucose for breakdown of energy. Respiration is the process of breaking down glucose to release energy. This process takes place in the cells of the body. Glucose is a sugar that is the body's main source of energy. When glucose is broken down, it releases energy that can be used by the cells to carry out their functions. ATP, energy carrying molecule. The energy released from respiration is stored in a molecule called adenosine triphosphate, ATP. ATP is the body's main energy currency. It is used to power all of the body's activities, from muscle contraction to cell division. Respiration in a cell. Respiration is the process by which cells break down glucose to release energy. 3. There are two main types of respiration, aerobic respiration and anaerobic respiration. Aerobic respiration takes place in the presence of oxygen. It is the more efficient type of respiration, and produces more energy. The first step of aerobic respiration is glycolysis, which takes place in the cytoplasm of the cell. In glycolysis, glucose is broken down into two molecules of pyruvate. Pyruvate is then transported to the mitochondria, where it is further broken down in the Krebs cycle and electron transport chain. The Krebs cycle produces carbon dioxide and water, while the electron transport chain produces ATP, the cell's energy currency. Anaerobic respiration takes place in the absence of oxygen. It is less efficient than aerobic respiration and produces less energy. The only type of anaerobic respiration that is shown in the image is fermentation. Fermentation takes place in the cytoplasm of the cell and produces ethanol or lactic acid as a byproduct. Glycolysis is the first step of cellular respiration, which is the process by which cells break down glucose to release energy. Glycolysis can occur in the presence or absence of oxygen. In the absence of oxygen, glycolysis produces two molecules of pyruvate, two ATP molecules, and two NADH molecules. The NADH molecules can be used to produce ATP in later steps of cellular respiration, but the pyruvate molecules cannot. In the presence of oxygen, pyruvate is transported into the mitochondria, where it is further broken down to produce more ATP. The enzymes that catalyze the reaction of glucose to pyruvate are called glycolytic enzymes. The two ATP molecules that are produced in the reaction are used to power the reaction itself. The two NADH molecules that are produced in the reaction can be used to produce up to 32 ATP molecules in later steps of cellular respiration. Anaerobic respiration produces less ATP than aerobic respiration because it does not produce NADH. NADH is used to produce ATP in later steps of cellular respiration. 4. The different pathways of glucose breakdown in a cell. The main pathways are aerobic respiration and anaerobic respiration. Aerobic respiration takes place in the presence of oxygen. It is the more efficient type of respiration and produces more energy. The first step of aerobic respiration is glycolysis, which takes place in the cytoplasm of the cell. In glycolysis, glucose is broken down into two molecules of pyruvate. Pyruvate is then transported into the mitochondria, where it is further broken down in a series of reactions that produce ATP, carbon dioxide, and water. Anaerobic respiration takes place in the absence of oxygen. It is less efficient than aerobic respiration and produces less energy. The type of anaerobic respiration is human muscles. Fermentation takes place in the cytoplasm of the cell and produces lactic acid as a byproduct. Cell division. Mitosis. It is a type of cell division that results in two identical daughter cells. The number of chromosomes remains the same in the daughter cells as in the parent cell. It is used for growth and repair of the body. Meiosis. It is a type of cell division that results in four daughter cells. The number of chromosomes is reduced by half in the daughter cells compared to the parent cell. It is used for the production of gametes, sex cells. The process of cell division in a human cell. Human cells have 46 chromosomes, which are made up of DNA. Meiosis is a type of cell division that produces gametes, or sex cells. In meiosis, the number of chromosomes is reduced by half, so each gamete has 23 chromosomes. This is because a human cell has 23 pairs of chromosomes, for a total of 46 chromosomes. In meiosis, each chromosome pair separates, so each gamete only gets one chromosome from each pair. Male cells produce sperm cells, which are the male gametes. Female cells produce egg cells, which are the female gametes. When a sperm cell fertilizes an egg cell, the two gametes fuse together to form a zygote. The zygote has 46 chromosomes, the same number of chromosomes as a human cell. This is because the sperm cell and egg cell each contributed 23 chromosomes to the zygote. Cell division is used to repair cells that have been damaged or injured. For example, if a person gets a cut, the cells in the cut area will divide to repair the damage. This process is called a representative cell. Types of cells. Somatic cells and gamete cells are the two main types of cells in the human body. Somatic cells make up all of the tissues and organs in the body, while gamete cells are the reproductive cells that produce offspring. Somatic cells have 46 chromosomes, which are arranged in 23 pairs. These chromosomes are made up of DNA, which is the genetic material that determines a person's traits. Gamete cells, on the other hand, only have 23 chromosomes. This is because when a sperm cell and an egg cell fuse together during fertilization, the resulting zygote has 46 chromosomes, the same number as a somatic cell. 5. The process of cell division that produces gamete cells is called meiosis. Meiosis is a more complex process than mitosis, the process that produces somatic cells. In meiosis, the chromosomes are paired up and then separated. This results in four daughter cells, each with 23 chromosomes. Somatic cells are essential for the growth and repair of the body. They also play a role in many other important processes, such as immunity and metabolism. Gamete cells are essential for reproduction. They are responsible for passing on genetic information from parents to offspring. Mitosis division. Nuclear division is the process by which the nucleus of the cell divides into two daughter nuclei. This happens in the M phase of mitosis, which is the stage where the chromosomes are duplicated and the cell prepares to divide. The chromosomes are duplicated in the first stage of mitosis, called the prophase. In the second stage, called the metaphase, the chromosomes line up in the middle of the cell. In the third stage, called the anaphase, the chromosomes separate and move to opposite poles of the cell. In the fourth and final stage, called the telophase, the nuclear envelope reforms around each set of chromosomes and the cell divides into two new cells. In the prophase stage, the chromosomes condense and become visible. The nuclear membrane breaks down and the nucleolus disappears. In the metaphase stage, the chromosomes line up in the middle of the cell. In the anaphase stage, the chromosomes are separated and move to opposite poles of the cell. In the telophase stage, the nuclear membrane reforms and the nucleolus reappears. The cell then divides into two daughter cells. The cell cycle. The cell cycle is a cyclical process that cells go through in order to grow and divide. The cell cycle has 4 main phases: G1, S, G2, and M. G1. This is the first gap phase, where the cell grows and prepares for DNA replication. During this phase, the cell increases in size and synthesizes proteins and RNA. S. This is the synthesis phase, where the DNA is replicated. During this phase, the cell duplicates its DNA so that each daughter cell will have a complete set of chromosomes. G2. This is the second gap phase, where the cell prepares for mitosis. During this phase, the cell synthesizes more proteins and RNA and makes sure that the DNA has been replicated correctly. 6. M. This is the mitotic phase, where the cell divides into 2 daughter cells. During this phase, the chromosomes condense, the nuclear membrane breaks down, and the mitotic spindle forms. The mitotic spindle is a structure that helps to separate the chromosomes during mitosis. The chromosomes then separate and move to opposite poles of the cell, and the cell divides into 2 daughter cells. 3. Parent baby. Mitochondrial inheritance is the process by which mitochondria are passed on from the mother to the child. Mitochondria are tiny organelles that are found in the cytoplasm of cells. They are responsible for generating energy for the cell. Consider a mother cell and a father cell. The mother cell has 2 mitochondria, one with damaged DNA and the other with normal DNA. The father cell does not have any mitochondria. When the mother cell and the father cell fuse together to form a zygote, the zygote inherits 2 mitochondria from the mother cell. The zygote then undergoes mitosis to form 2 daughter cells. The 2 daughter cells will each inherit 1 mitochondria from the mother cell. If the mitochondria with the damaged DNA is inherited by the daughter cell will not be able to generate enough energy and will die. This is a rare condition, but it can happen. In some cases, the child may be born with a mitochondrial disease. Mitochondrial diseases are a group of disorders that are caused by defects in mitochondria. These disorders can affect many different parts of the body, including the heart, brain, and muscles. 7. Mitochondrial diseases are caused by mutations in the DNA of mitochondria, which are the energy-producing organelles found in cells. These mutations can be passed on from mothers to their children, but only if the mother's egg cell contains mitochondria with the mutation. In a 3-parent baby, the mitochondria from the mother's egg cell are replaced with mitochondria from a healthy donor. This is done by removing the nucleus from the mother's egg cell and then fusing it with the cytoplasm of a donor egg cell. The resulting cell, which has the mother's nucleus and the donor's mitochondria, is then implanted in the mother's uterus to grow into a baby. The different steps involved in 3-parent baby. The first step is to remove the nucleus from the mother's egg cell. This is done by using a laser to cut the cell in half. The nucleus is then removed from one half of the cell. The second step is to remove the cytoplasm from the donor egg cell. This is done by using a suction device to remove the cytoplasm from the cell. The third step is to fuse the nucleus from the mother's egg cell with the cytoplasm from the donor egg cell. This is done by using an electric current to join the 2 cells together. The fourth step is to implant the resulting cell in the mother's uterus. If the cell implants and grows into a baby, the baby will have the mother's nuclear DNA and the donor's mitochondrial DNA. Q. In the context of hereditary diseases, consider the following statements, 2021. 1. Passing on mitochondrial diseases from parent to child can be prevented by mitochondrial replacement therapy either before or after in vitro fertilization of the egg. 2. A child inherits mitochondrial diseases entirely from the mother and not from the father. Which of the statements given above is correct? A. 1 only. B. 2 only. C. Both 1 and 2. D. Neither 1 nor 2. Surrogacy. Surrogacy is a complex process in which a woman agrees to carry and deliver a child for another couple or individual. The process typically involves the following steps. The intended parents, IPs, provide their sperm and egg cells to a fertility clinic. The clinic fertilizes the egg cells with the sperm cells in a laboratory. The resulting embryos are transferred to the surrogate's uterus. The surrogate carries the pregnancy to term and delivers the baby. The IPs take custody of the baby after the birth. Blood. The different components of blood are labeled. Components of blood. The different components of blood, including red blood cells, white blood cells, platelets, and plasma. 8. Red blood cells carry oxygen to the tissues. White blood cells fight infection. Platelets help blood clot. Plasma is the liquid part of blood that carries nutrients and waste products. Plasma. Plasma makes up about 55% of blood. It is a clear, yellowish liquid that contains water, proteins, nutrients, and waste products. Hemoglobin. Hemoglobin is a protein found in red blood cells. It is responsible for carrying oxygen to the tissues. Red blood cells, erythrocytes. Red blood cells are the most abundant cells in blood. They are disc-shaped and contain hemoglobin. These are the cells that carry oxygen to the body's tissues. They make up about 45% of blood. Red blood cells are red because they contain hemoglobin, which is a protein that binds oxygen. White blood cells, leukocytes. White blood cells are less abundant than red blood cells. They are there to fight infection. They make up about 1% of blood. White blood cells come in different types, each with a different function. Platelets, thrombocytes. Platelets are the smallest cells in blood. They help blood clot. They make up about 1% of blood. Platelets are small, disc-shaped cells that help blood clot by forming a mesh that traps blood cells. Blood has the following features. It is fluid. It is red. It is made up of different components. It is essential for life. Note: the one hemoglobin molecule carries 4 molecules of oxygen. Human beings have 5.5 liters of blood in the body. White blood cells, WBCs. The different types of WBCs are granulocytes. These WBCs have granules in their cytoplasm. They include neutrophils, eosinophils, and basophils. Neutrophils. These are the most abundant type of WBCs. They fight infection by engulfing and destroying bacteria. Eosinophils. These WBCs help fight parasitic infections and allergic reactions. Basophils. These WBCs release histamine, which is a chemical that causes inflammation. 9. Agranulocytes. These WBCs do not have granules in their cytoplasm. They include lymphocytes and monocytes. Lymphocytes. These WBCs fight infection by producing antibodies. Monocytes. These WBCs mature into macrophages, which are large cells that engulf and destroy bacteria and other foreign invaders. Lymphocytes are a type of white blood cell that helps the body fight infection. They do this by producing antibodies, attacking infected cells, and killing cancer cells. There are 3 main types of lymphocytes: B cells, T cells, and natural killer cells. B cells produce antibodies, which are proteins that help the body fight infection. When a B cell encounters an antigen, it will produce antibodies that are specific to that antigen. These antibodies will then attach to the antigen and help the body's immune system destroy it. T cells attack and kill infected cells. There are 2 main types of T cells: helper T cells and cytotoxic T cells. Helper T cells help other cells of the immune system, such as B cells, to function properly. Cytotoxic T cells kill infected cells directly. Natural killer cells attack and kill infected cells and cancer cells. They do this without the need for antibodies. Memory cells are a type of lymphocyte that remembers the specific antigens that the body has been exposed to in the past. This allows the body to fight infection more quickly if the same antigen is encountered again. The process of the immune system fighting infection. When the body is invaded by a foreign invader, the white blood cells recognize the antigen on the surface of the invader. The white blood cells then produce antibodies that bind to the antigen. The antibodies attach to the antigen and mark it for destruction.